Hey, Mr. P here. In this video, we're going to talk about the kind of in-depth biology behind muscle contraction. In previous videos, we did kind of an overview of muscle contraction. We talked about the kind of mechanisms from an anatomical standpoint, bones, tendons, ligaments, elbows, joints, that kind of thing. And now we're going to move into the actual level of organization that is muscle tissue and muscle cell tissue specifically, and really kind of talk about why a muscle tissue is structured the way it is and how that structure allows it to function the way that it does. Muscle tissue cells are incredibly adapted to not only utilize a variety of different environmental outcomes or environmental conditions, but they actually allow organisms to do a variety of different things which are needed in order for those organisms to kind of live within the environments that they've become accustomed to living. So, what is a striated muscle cell? A striated muscle cell is a cell that contains a variety of different cellular components like other eukaryotic cells do, but they are highly specialized to perform a highly special function. And that function is all about contraction. And contraction shortens muscle fibers so that they can pull on levers, which are bones, in order to move an organism. The first thing that I like to point out is that, like in previous topics, specifically cell biology, we talked about how muscle fibers are kind of an exception to the cell theory. And we talked about that muscle cells are multinucleated, meaning, and it's not really depicted in this particular picture, but when you look at muscle cells, they actually have a bunch of different nuclei that are right inside the membrane and can typically be seen kind of as structures that look like nuclei, but there are a ton of them, meaning many, multiple, okay, plural, which is an exception to the way cells have always been structured, because we've always talked about eukaryotic cells only possessing one nucleus at most. So muscle cells are multinucleated. That means they have multinuclei. They are always positioned along the exterior, but along the edges, and can actually most of the time be seen through the membrane that is the sarcolemma. Okay. Some other specific things that you need to be able to identify within a striated muscle cell are the mitochondria. There are a bunch of mitochondria. You can see them. They're all in here. They're between these microtubules or the myofibrils. Basically the bundles that make up the muscle tissue cell, and that's because there is a huge ATP demand that is placed on these cells because of the fact that organisms are moving all the time, and muscle contraction requires ATP. You cannot contract or relax a muscle without ATP, and the only way to produce ATP in an aerobic organism is through aerobic respiration, which is obviously done at the level of the mitochondria. These muscle fiber cells are held together by a plasma membrane, which is specific to the muscle fiber or the muscle cell, and it's called sarcolemma. Okay, and that sarcolemma is a specialized cell membrane that holds all of this structure, and the sarcoplasma, which is kind of like the cytoplasm, into or inside this cell. We'll talk more about these particular structures in a minute. There are what is called T-tubules or transverse tubules. Those T-tubules are little invaginations or little projections that protrude from the sarcolemma, which is the cell membrane, into the interior of the cell. And so it's these invaginations that make up these T-tubules. It's a way to get materials in and out of the cell more efficiently. There also is sarcoplasmic reticulum, which may sound very similar to the endoplasmic reticulum. The sarcoplasmic reticulum is a specialized type of ER that stores calcium ions specifically. It also helps to pump them in and out of the sarcoplasm, which is, just like I said, the cytoplasm of these particular cells. And we will talk about what the role of those calcium ions is in a minute, but you cannot contract a muscle without ATP and without calcium ions. So that sarcoplasmic reticulum and the muscle cell's ability to store high amounts or high concentrations of calcium ions is going to be critical when we go to actually talk about how the muscle contracts. And then we have these myofibrils, which are these actual bundles, much like a vascular bundle within plant tissue, to move structures or to move water and flow them in and out of the plant. These are basically protein-based contractile units that are grouped together and known as muscle fibers. So when you look at a muscle, you're going to notice that it's very stringy. You're going to notice that it's very fibrous, and all of those fibers are protein-based, and they all have the ability to contract. And like I said, the whole purpose of this video is to talk about not only why they're fibrous and why they're filamentous, but also how the fibrous nature of that particular cell, gives it the ability to contract. Myosin and actin are two types of proteins. Actin is a small protein complex, and myosin is a thick protein complex. So you'd see that there's a thin myofilaments and there are thick myofilaments. The thick ones obviously look darker when you look at them under a microscope. The thin ones obviously don't look as dark because they're not as thick. But myosin and actin are the actual functional proteins that make muscle contraction possible, and we're definitely going to talk about those in detail in a minute. Okay. So if we kind of zoom out, obviously we're talking about skeletal muscle. There are other types of muscle in your body, specifically like cardiac muscle, there's smooth muscle, longitudinal muscle, but we're going to focus on how skeletal muscle helps to contract and therefore move body parts within an organism. And these skeletal muscles, basically the muscles that make up your muscular system, are made up of these bundles called myofibrils, which we can see here, which are made up of actin and myosin, which we already talked about. Okay, actin being these really thin protein complexes or protein bands, and the myosin with these little kind of projections. It makes them look kind of rough. Those are the myosin, and those are thick. And so you can see that there is actually a difference between the thin and thick bands of these particular muscle fibers. But you'll also see that there's a Z-line, or what we call a Z-line, and another Z-line here. And so Z-line to Z-line makes up one functional unit of the muscle cell, which we call a sarcomere. Okay, so sarcomeres are the repeated structural unit of muscle cells, and they basically travel from Z-band to Z-band. Now you'll also see that there are different bands within kind of a sarcomere, and there are I bands and A bands. The A bands make up these really thick, dark bands because that's where all of the myosin is kept. And then you have the I bands, which is kind of lightly colored, and that's because that's where just the actin is kept. Okay, and again, we're going to talk more about this in a minute. We're kind of just laying the foundation for muscle contraction, and then we'll move on in a minute. Okay, so repeated unit of actin and myosin help to make the sarcomere, and the sarcomere is the functional unit of contractile muscle fibers, which gives us the ability to move. Now, if we focus in on an individual sarcomere, again, a sarcomere goes from Z-band or Z-line to Z-line. There is a really thick group of proteins called myosin. The myosin have these little hand projections, okay, or foot projections that extend off of the surface of the myosin, which are going to be critical when we talk about how the muscle contracts in a minute. You're also going to have these actin, which are thin and don't have these little kind of projections that are protruding off of them. And so when you look at a muscle fiber from sarcomere to sarcomere to sarcomere, you will notice that there is always a section that is kind of dark, okay, which again is the A band. And then you have a section that is light, which is only containing the actin, and those make up the I band. Okay. Now there are some other things on here like C protein and M line and the creatine kinase. Those are enzymes that help facilitate proper mechanics and mechanism of muscle contraction. We're not going to worry about those. You need to know though some terms like A band, I band, Z line, actin, myosin, stuff like that. Okay? So, how does a muscle actually contract? Now, they're much like other biological processes. There are steps that need to take place, and the steps are 1, myosin heads, which are these little projections that I kind of alluded to on that previous slide. Okay, the myosin, that is the thick filament, and they have those little projections. We call them myosin heads. They are activated by splitting an ATP. Now, the ATP in this particular diagram has already been split. And we know that it's already been split because when ATP splits, it splits up into two kind of products, right? ATP splits and it forms an ADP and it forms a inorganic phosphate. When ATP splits, it activates the myosin head and actually causes the ADP and inorganic phosphate to bind to the myosin head, which causes the myosin head to actively bind or grab a hold of the actin at these myosin binding sites, okay? Myosin head binding sites. You can see that they're the little kind of circles or the little, what look like little slots that these heads can bind to. Again, we're going to dive in on the next slide, and we're going to talk more about those myosin binding sites in more detail. But for now, this myosin head has to bind to those particular myosin binding sites that are located on the actin, okay? Once the myosin head binds to those actin myosin binding sites, it's going to basically release the inorganic phosphate and ADP. They're going to jump off because now that myosin head is bound to the actin. And if it's bound to the actin, it no longer can bind to the inorganic phosphate and ADP. So this inorganic phosphate and ADP is released, okay? It is releasing those two reactants or those two products from this initial ATP being split up or broken down. As the myosin forms the cross bridge, okay, which is the bridge between the myosin and the actin, they're actually physically linked now. The myosin bends due to the loss of energy, and the reason it lost energy is because it lost the ADP and the inorganic phosphate, which still has some energy in it. I mean, ADP is a low energy molecule, but it still contains energy. The bending towards the center of the sarcomere, and therefore the actin is moved inwards. And so what happens is, do you see how this is kind of like angled kind of out to the right? Well, it's going to grab a hold and it's going to physically bend and snap this way. And when it does, it actually pulls this entire actin filament this way, which if you look over here, and if you pull the actin this way, you're actually going to pull this Z line closer to this Z line, which means your muscle fiber as a whole is getting shorter. That is the definition of contraction. When you contract a muscle, the muscle fiber actually gets shorter, meaning if you want to bend your arm, you have to contract the muscle that causes the lower arm to get basically pulled towards your upper arm, and it causes it to bend. That would be the bicep. Okay, then myosin bends to or binds to ATP, and so now it's going to detach from actin, no longer is attached to these myosin binding sites. It's going to attach to an ATP, and it's therefore going to split the ADP from inorganic phosphate again, and those are going to stay bound, and as long as those two things are bound to the myosin head, it's no longer going to bind to that actin, and it's going to cause the myosin and actin to release, and therefore can actually disallow the contraction, and it can cause the muscle to relax. So I said that we would dive into the actual myosin binding sites. This is the way that tropomyosin and troponin are actually kind of embedded within the actin filaments. The actin filaments, they are really important for us to track, okay? This entire thing is an actin filament, and on the surface of the actin filament are these really, really thin proteins called tropomyosin. The purpose of the tropomyosin is to completely block these myosin head binding sites so that the myosin doesn't bond to them and therefore keeps the muscle relaxed. Because if you don't bind the myosin heads to the myosin binding sites, you can't contract the muscle and it stays relaxed. Troponin is a protein complex that actually is bound to the tropomyosin every so often, and its job is to actually bind to calcium ions. Remember in that previous slide, or earlier in the video, I talked about the importance of calcium ions. those calcium ions are going to be released by the sarcoplasmic reticulum, and I said that the calcium ions are vital for the proper contraction of a muscle. And so what's going to happen is when a muscle is not contracting, the binding sites on the actin are covered by a thin protein called tropomyosin. Okay, we've already established that. Tropomyosin sits here and it blocks those myosin head binding sites so that myosin can't bind to the actin. Troponin is a protein complex that binds to the tropomyosin at regular intervals. Okay, it's here, and then it's here, and then it's here, and then it's here. And the purpose of that is to actually have or contain binding sites for calcium ions. When the sarcoplasmic reticulum releases those calcium ions, meaning we want to stimulate a muscle contraction, those calcium ions are going to actually bind to the troponin. And when those calcium ions bind to the troponin, it's going to cause the tropomyosin to slide off of those myosin binding sites. And when it does that, it exposes them so that the myosin heads can then bind to these myosin binding sites. And when they bind to the myosin binding sites, it allows the myosin to start moving the actin filaments and bringing those Z lines in towards each other, which shortens the muscle fiber, which contracts the muscle. Okay? Instantly, the myosin heads find an actin binding site and undergo the flex movement of the myosin heads, leading to the shortening of the sarcomere and thus the entire muscle. A muscle that is contracted is a muscle that is shortened, and a shortening muscle or a muscle that has been shortened is a muscle that is contracting. Okay, you have to know that. A muscle that is relaxed is a muscle that is not shortened. It's elongated, right? A relaxed muscle is a long muscle. A short muscle is a contracting muscle. Okay? Here we go. Muscle contraction explained. For all of you individuals that are better off reading kind of step by step by step, I tried to order these in a series of events that give you kind of a detailed explanation of exactly what is happening in order to successfully contract a muscle. Okay? And so we start with the fact that muscle fibers contain repeated units called sarcomeres. We've already established that. Okay, the sarcomere is a functional unit that is Z-line to Z-line. Now, there are actual myosin, which are thick, and then there's an entire structure of actin that goes between them. And so that particular sarcomere, which is that Z-line to that Z-line, is the functional unit that will actually shorten when it is time to contract the muscle. Okay? So, first thing that has to happen is a motor neuron carries an action potential. Now, we haven't really talked about action potentials or the neurons in previous videos, but we will. The action potential has to reach the final synapse, which is the end of the neuron, and actually terminates at a neuromuscular junction, which means the brain sends a signal from brain down the neuron and it terminates at the muscle. When it terminates at the muscle, it releases a neurotransmitter called acetylcholine. Acetylcholine is released into the synaptic gap, which is the gap between the neuron and the sarcolemma, which we remember is the actual membrane that surrounds or encases the entire muscle fiber or the muscle cell. When the acetylcholine is reached by the sarcolemma, the acetylcholine that was released is going to bind to receptors on the sarcolemma, which is the membrane of that muscle fiber. And when it binds to the receptors, it's going to open up the ion channels and release sodium ions through the membrane, which are going to cause an action potential to move through those T tubules. Remember, those are the transverse tubules that are invaginating or invaginations on the inside of that sarcolemma. And that causes the release of calcium ions from the sarcoplasmic reticulum, which is where they're stored, which is going to cause a substantial increase of calcium ions in the sarcoplasm, which is going to do what? It's going to cause those calcium ions to flood into the sarcoplasm and therefore cause the calcium ions to bind to the troponin, which is that protein complex that is located on the tropomyosin. When the calcium ions bind to the troponin, it's going to cause the tropomyosin to slide off of the myosin binding sites, which is located on the actin. When the myosin binding sites are exposed, the myosin heads will attach to those binding sites on the actin and will again start the contraction process. The myosin heads will all flex towards the center of the sarcomere, and while they are flexing towards the center of the sarcomere, the entire sarcomere shortens as the Z lines move towards each other. And as the Z lines move towards each other, they are physically getting shorter, which means the muscle is contracting. ATP then binds to the myosin head, and as ATP binds to the myosin head, it detaches the myosin head from the actin so that it can await another action potential from a motor neuron, which starts this cascade of events again, which will either cause the sarcomere to continue to shorten if the neurons are telling or are sending signals to continually contract, or they will slide the other way and they'll completely lengthen if the neurons are carrying signals from the brain which are telling the muscle to relax. Okay, sarcomere shortens as a result of sliding filaments. That is the sliding filament theory, and this is the way that muscles contract and how muscle contraction works. I've seen plenty of IB exams in which they ask you an 8-mark question centered around muscle contraction. As you can see, there are more than 8 bullet points here because there are a variety of different bullets or different items that can be included in your response, which will give you the points for those for that question. The last skill that we need to kind of talk about is identifying different aspects of muscle contraction or different stages of muscle contraction from electron micrographs. And so you can see that there is a series of micrographs here. This is showing you on the left side of the screen the difference between fully relaxed muscle fibers and fully contracted muscle fibers. Hopefully you can draw some conclusions. The first conclusion I want you to draw is that when you look at the dark band, they are always the exact same width, regardless of if the muscle is contracted or not. The dark band is the same width always. The difference between a fully relaxed muscle and a fully contracted muscle is how wide the light band is and how close the Z band or the Z lines are. Okay? In a fully contracted muscle, this Z line, which is here right in the middle of the light band, is actually pulled in closer to the center of the dark band. And so if we were to measure this distance from Z line to Z line, that is a shorter distance than this Z line to this Z line. This is a longer distance, okay? Longer distance equals more relaxation. Shorter distance equals more contraction. Again, if you look at all of these on the right side of the screen, you can see that they are kind of shown side by side by side by side. Again, the dark band is exactly the same width in every case. The difference exists in where exactly the Z line is and how close the Z line has been pulled to the dark band. As these Z lines get pulled in closer to the center, the muscle is in more and more and more contraction. And as those Z lines back out, it's more and more and more relaxed. Okay? If you have any questions, I know this video is a really in-depth look at some HL content, but if you have questions, bring them to class. See ya.